Hi, I'm back. So let's think about this. So now we're on chapter 6, part 2. And we're going to talk about bacterial cell division first. So think that every generation you're increasing the number of cells as they're growing, right? Bacterial cells. So if you get first generation 0, start with generation 0. You have 1 cell. Generation 1 cell splits into 2 by binary fission, right? We're going to show the process of binary fission in a minute. Generation 2, you have 4 cells. Generation 3, you have 8 cells. Generation 4, 16. You see how you're just elevating 2 to the generation will give you the number of cells per generation. 2 elevated to 5, meaning 5 generations, you have 32 cells. Okay? What am I trying to tell you here? I'm trying to tell you that, let me just show you the binary fission process, right? One bacterium, that's the bacterial genetic material inside the nucleoid. Bacteria divides and splits in 2. That DNA was already duplicated, splits, half goes to each one of the cells. 1 mother cell, 2 daughter cells, okay? So when you look at a growth curve, what is important to know here? If I plot the numbers from this table here, do you agree that the first 5 generations are going to be really, really parallel to the x-axis? Because it's a tiny number compared to when you get to like 10 generations, 15 generations. Look at 17 generations. Look at 20 generations. It's a log growth, right? Because you're elevating to that generation number. That's how they grow. So that means for you to be able to see these numbers when you have 5 generations, it's really hard to see when you're plotting the graph like this. But if you multiply these numbers by a log of 10, then you have a straight line. You see? So that's why, because this growth is exponential, we apply a logarithm scale to be able to see them. Okay? So now here it's more comparable. The amount how the growth is constant between the 1st and the 5th generation, between the 10th and the 15th generation. It's a constant growth. And you can see that because you put in a log scale. Okay? Phases. Growth of bacteria happens in these 4 distinct phases. So you start with the lag phase. What is the lag phase? Excuse me. It's a phase where bacteria are not really growing. They're just preparing to grow. So there is an intense metabolic activity for them to start multiplying, but nothing is really happening when it comes to the number of cells. So log of number of bacteria is constant in the lag phase. Log phase. Everybody's growing exponentially, just like we showed in the previous graph, right? Everybody growing crazily. What's the stationary phase? It's the phase where the number of bacterium growing equals the number of bacterium that starts to die. Meaning, growing a lot, but also dying a lot, it kind of cancels themselves. So you have a constant number of cells here. Even though you have bacteria growing, you have bacteria dying. So the number is steady, it's constant. And then the death phase, which you know what happens, right? So these are the 4 phases that I want you to keep in mind. So now let's talk about measurements of microbial growth. We have different direct methods, which are plate count, filtration, MPN, and direct microscopic count. In your learning objectives, I said focus on serial dilution, plate count, filtration, and the microscopic count on chamber. So we're not going to talk about MPN, okay? Plate count. So what is this? You're counting colonies on plates that have between 30 and 300 colonies. So you dilute serially. You're going to get your original inoculum, and you're going to dilute many different times in series. So you dilute the first, and from the first you get a little bit and add to more liquid. From the third or the second, you get a little bit and dilute again. And why are you diluting all these tubes? Because when you do serial dilution first, if there is an error, you keep that same error because it's one depends on the other. Second, you're going to plate all these dilutions, a specific volume, right? You're plating like 1 ml in this plate, because you don't really know which one of these plates is going to give you between 30 and 300 colonies. Remember, you need between 30 and 300 colonies to avoid major errors, okay? So then what you do is you count the bacteria here, and then you just divide by the dilution multiplied by this volume, and you have the cell density. Filtration. Filtration is a method where you pass the solution that contains the bacteria through a filter, and you collect the bacteria from the filter. So this is an example of a filter with the bacteria on top of it. And another example here. So then what you do is you get that filter and you put it like upside down on a medium and you let it grow. And this is what happens. Then everywhere where there was a little group of bacteria or one single bacteria is going to form a colony. And then you count the colonies. So filtration is used for when you have a low density of bacteria in your sample. And then you filter and you're concentrating them, and then you plot them, right, upside down in the agar medium, and then you let them grow. So the difference between filtration and serial plate count, serial dilution and plate count, is that here you have a low concentration of bacteria in your sample. In the previous one, in the plate count, you have a high concentration and you need to dilute it to be able to count, and then multiply back to get to the original inoculum concentration of bacteria. There is another method that is direct also, that is just you add your sample, diluted or not, to a chamber. This chamber has these little grids, and there is a specific number that you multiply where that is related to the depth and the area of this chamber. And multiplying by this number, you count this bacteria. These are not colonies. These are just bacteria. You're looking under the microscope. You count them, multiply by a specific number that I'm not really going to ask you details, but there is a factor that you multiply to get to how many cells per ml you have. Okay? So this is the chamber counting method. Indirect methods. So for indirect, I wrote in your learning objectives, we're going to talk about turbidity. That's the only one. So what is turbidity? I don't know if you ever noticed that when bacteria grow in a tube, in a sample, in a juice, in whatever medium that the bacteria is, and it grows and it's liquid, it gets cloudy, right? And that cloudiness is what we call turbidity, right? So a turbid medium is a cloudy medium. So when you measure that cloudiness with a device that is called, where's my pen? That is called spectrophotometers. These devices are able to measure the cloudiness of the medium by measurement that is called absorbance. I'm going to write here because I think it's not in the slide. Absorbance, right? So measuring the absorbance with the use of this type of equipment, spectrophotometer, will give you the cell density, right? The more bacteria, the more cloudy, the more absorbance you can measure. Does it make sense? And then you have to just apply a formula to know exactly how many bacteria you had. Cool? So I told you about microbial growth and ways to measure this growth. Next chapter, we're going to talk about how to control microbial growth, which is something that we are very interested in when it comes to clinic, right? So I see you for chapter 7. Bye-bye.